Hello students, welcome back to my channel. We are learning ICSE 10 biology. We have been learning all the important chapters till now. We are in transpiration, and in the previous video we looked at how to demonstrate or show transpiration occurs. We proved that. Now we know that transpiration occurs, but how to measure how much transpiration is occurring? For that, we have few experiments given in the textbook. Let's learn all of them. Till now, if you haven't subscribed to my channel, please do, so that you can share your questions with me, your doubts with me. You can learn along and score full marks in biology in the board exam. Okay, the first method given here is weighing method. What is weighing method? You have to weigh something. What are we weighing? First, you take a potted plant and then take the weight of it. First step. Second step, keep it in the sunlight. Before keeping in the sunlight, the bottom part of the plant, that is where the soil is, where the pot is, that you need to cover and then keep it in the sunlight. Why we need to cover that? To prevent evaporation. Okay, then weigh it. After taking the weight, you will notice that the weight has reduced because of loss in water, proving how much water has transpired. First one. Second one, similar thing we are doing, but in a different bottle. Okay, in a bottle you're taking a plant. Make sure that there is no air gap here by covering it using a cork. This bottle is connected to a tiny graduated tube which is also partially filled with water. Take the weight of it. Step number 1. Step number 2 would be keeping the setup in sunlight. Step number 3 is again weighing it. Take the weight of it. You will see that the weight has reduced because of transpiration. Here you are saying one more thing that is similar to this experiment where we did double visual proof we did, right? Here you saw the water droplets also, you saw change in color also. Like that, here two things you will notice. One is loss in weight, that is lesser weight compared to initial weight. Second is water level will come down in this tube. Why water level will come down? Because due to transpiration or loss of water from the leaf, the root will absorb water. Therefore, here water level will come down. So, two things you are doing with the second experiment. Third is very similar to it. Instead of the entire plant, we are taking a small shoot. And in a test tube you are doing, before and after keeping in the sunlight, you are checking the weight. Same thing, but here you will easily observe much loss in water because this is only a small twig. Okay. Now, one more thing is, why is oil used here? Same question we had done previously also. Oil is used to prevent evaporation of water. Next one important part in this chapter is Potometer. Very, very important. Definitely there is one question from here. What is a potometer? What are the three types? How does it function? What is the limitation? Any one of this part, you are getting 100% one question. So what is a potometer? It is a device to measure, please underline, the rate of water intake by the plant. Okay. So therefore, indirectly, you can measure transpiration. What do I mean by that? You have a setup like this. Okay. This is the potometer, this bend tube. In that, one side of it, you are dipping in a beaker with colored water. Other side, you are keeping a twig, a stem kind of part with lot of leaves on it. There is a reservoir to fill water, and the entire setup you are filling with water. First step. Second step is this part you have to lift up and make sure one air bubble gets inside this. Once that is done, put it back in such a way that not too many water bubbles enter. Water bubble will slowly start moving up and it will wait here. Meaning it would be stuck somewhere in this tube. You're keeping the entire setup in sunlight. What happens when you keep it in sunlight? From the leaves, transpiration will occur. Water will be lost from here due to adhesion, cohesion, all the forces that help in ascent of sap. Water will be pulled up from here. Correct? Therefore, this water here will move up. Slowly, air bubble will start moving towards this side. How much this air bubble moves is an indirect indication of how much water has transpired. But you are not checking directly transpiration. What are you indirectly checking? Directly you are checking water intake, rate of water intake. And indirectly you can measure transpiration. Please underline those 2 points. Limitations is another favorite question in board paper. Any two limitations of the potometer is definitely asked. Please study. We'll continue learning other parts of the transpiration and kinds of transpiration in the next video. But I would like to explain just one more experiment that is there in the text. Here, what are we proving? Aim is very important to learn. Okay. What are we learning and what are we proving? We are proving that the amount of transpiration on upper surface and lower surface of the leaf are at different levels. Why is it different? Definitely you know the reason. On the lower surface of the lower surface of the leaf, more stomata are present, more stomatal openings are present in the lower side. Therefore, more transpiration from the lower side. Upper surface, very less amount of stomata are present, therefore less transpiration occurs. That you know. But how to prove that? To prove that, again, we need our cobalt chloride paper, which is blue in color generally. What do? We take the leaf. On the leaf, we keep a cobalt chloride, small strip of cobalt chloride paper. Then you keep slide. You know what is a slide? You in the biology lab, you must have seen a slide. Okay, glass slide. On top of the cobalt chloride paper, you're keeping the glass slide. On the bottom of the leaf also, you're keeping the glass slide, but not the cobalt chloride paper in the first setup. In the first setup, on the top surface of the leaf, you're keeping cobalt chloride paper. On both sides, you're keeping the slide and putting a clip. Again, in the second setup, on the leaf, down part you're keeping the cobalt chloride paper. Slide you're keeping both up and down so that you can clip it. Alright, now after some time you will notice that water would have come out due to transpiration from the lower surface more, turning the cobalt chloride paper into pink. Why? Because it indicates moisture. I hope this is clear. On the upper surface, there will not be much of a change because very less transpiration, so less that it is not shown in the cobalt chloride paper. Okay, so that's what this experiment is. I hope it is clear. We will learn types of transpiration and how water moves or water comes out of the stomata. All those we will learn in the next video. Keep watching. Please do subscribe to my channel. Thank you.